Look at the blood type written on your medical record. Maybe it says O positive, a simple combination of one letter and one symbol that most people barely notice. In a hospital, it matters because it helps doctors determine which blood can safely be given to you. But biologically, that tiny label sits on top of a story that reaches far deeper than any medical form suggests. Your blood group is the product of ancient genetic variation, migration, population bottlenecks, and an evolutionary struggle against infectious disease that has been happening for thousands, and in some parts of the ABO system, millions of years. But there is one important thing to get straight before we go any further. O positive does not trace back to one mysterious ancestor, it does not prove that you belong to one ancient tribe, and it does not mean your blood is somehow superior. The real story is stranger than that. Type O exists because several genetic changes can produce an inactive version of the ABO enzyme, and those variants have been carried through populations across the world. Some of those variants became common because of population history. Others may have been shaped by disease. And in certain environments, the very blood group biology that can reduce the risk of severe malaria is also associated with worse outcomes from cholera. Your blood is not a secret code telling you exactly where your ancestors lived. It is something more interesting, an evolutionary record of the pressures they survived. To understand that record, we need to go back to Vienna more than a century ago, when a physician named Karl Landsteiner was trying to solve a medical problem that seemed almost impossible. Blood transfusion could save a dying patient, but doctors could not reliably predict when a transfusion would work and when it would trigger a catastrophic reaction. Landsteiner mixed blood samples from different people and noticed a pattern. Some combinations remained smooth. Others caused the red blood cells to clump together. That simple observation led to the identification of the A, B, and O blood groups, with AB recognized shortly afterward. Suddenly, doctors had a scientific explanation for why some transfusions were dangerous. Landsteiner's discovery transformed medicine and eventually earned him the Nobel Prize in Physiology or Medicine in 1930. But he had only solved the immediate medical mystery. He had not uncovered the evolutionary story behind the blood groups. That would take another century of genetics, population studies, and molecular biology. Today, we know that the ABO system is controlled by a gene located on chromosome 9. The gene encodes an enzyme that modifies a carbohydrate structure called the H antigen on the surface of red blood cells. The A version of the gene produces an enzyme that attaches one particular sugar. The B version produces an enzyme that attaches another. Common O alleles, by contrast, contain mutations that disrupt the enzyme, leaving the H antigen largely unmodified. The result is the O blood group phenotype. It sounds straightforward until you look closely at the DNA. There's not one universal O mutation shared identically by everyone with blood type O. Researchers have identified multiple O lineages with different evolutionary histories. One important O lineage carries a famous deletion that disrupts the enzyme, but other O variants arose through different changes. Genetic studies of archaic humans have found O-associated variants in Neanderthals, while genomic analysis has also identified O alleles in Denisovan ancestry. That means the genetic history behind O is not something that appeared recently in Europe, Asia, or the Americas. It reaches back before modern human populations became what they are today. One study of Neanderthal genomes concluded that the genetic change responsible for the O phenotype existed before the split between Neanderthals and the lineage leading to modern humans. Another analysis found typical O alleles in a Denisovan individual. So when you see O on a blood test, you're looking at the modern expression of a genetic system whose history reaches into deep human evolution. Now comes the part where geography enters the picture. Blood type O is common across many parts of the world, but in several indigenous populations of the Americas, it historically reached extraordinary frequencies. That pattern became an important question for geneticists. Was O somehow created in the Americas? Or had a population carrying high levels of O entered the continent already? Ancient DNA helped narrow the possibilities. Researchers examined pre-contact Native American remains and found that the ABO frequencies in ancient individuals from eastern North America were not significantly different from those seen in indigenous populations from the same region today. That strongly argues against the idea that the modern pattern was created entirely after European contact. The researchers concluded that a founder effect during the initial settlement of the Americas was the most consistent explanation, while also noting that genetic drift and natural selection could contribute. In simple terms, the first populations entering the Americas carried only a subset of the genetic diversity present in their ancestral populations. If O was common among those founders, later isolation could amplify it dramatically. This is what a founder effect can do. Imagine a large population containing several blood group alleles. Now imagine a small group breaking away and moving into a new territory. They do not carry the full genetic diversity of the original population. They carry a sample, Pure chance can make one allele unusually common in that small founding group. As their descendants multiply, that unusual frequency can become the normal pattern of the new population. If the population then remains relatively isolated, genetic drift can push the frequency even further. That is one reason you can get a population with an extremely high frequency of O, even though nearby or ancestral populations have a much more mixed distribution. The blood type is not proving where the people came from by itself. It is showing what happened to the genetic variation carried by a particular population, and this is why your blood type cannot be used as an ancestry test. A person from South America and a person from Europe can both be O positive. A person with mostly one ancestry can carry the same ABO phenotype as someone whose ancestry comes from several continents. One gene does not contain your complete migration history. Modern ancestry analysis depends on thousands of genetic markers spread across the genome. Blood type is just one small piece of that puzzle. What makes it fascinating is not that it can identify you. It is that population-level patterns and blood group frequencies can preserve evidence of ancient demographic history. But if the ABO system were only about inheritance and chance, the story would already be interesting enough. What made researchers really pay attention was disease. Blood group molecules sit at interfaces that pathogens encounter. That means microbes can sometimes exploit differences between people with A, B, AB, or O blood. Over evolutionary time, this creates an opportunity for natural selection. A genetic variant that changes the success of an infection can influence who survives, who reproduces, and which blood group frequencies become common in future generations. Scientists have therefore spent decades asking whether epidemics helped shape ABO diversity. The evidence does not point to one single disease responsible for the whole system. Instead, it suggests a complicated balance in which different pathogens may favor different blood groups. Malaria offers one of the strongest examples. The most dangerous human malaria parasite, Plasmodium falciparum, infects red blood cells and changes their surfaces. Those infected cells can bind to one another in structures called rosettes and can attach to blood vessel walls. Excessive rosetting and adhesion contribute to blocked circulation in severe disease. Clinical and genetic studies have found that people with blood group O tend to have a lower risk of severe falciparum malaria than people with non-O blood groups. The protection is not absolute. O does not make a person immune to malaria, and it does not prevent infection. The association is specifically strongest for severe disease. But in regions where malaria has been a major cause of childhood illness and death for countless generations, even a modest survival advantage could matter evolutionarily. Picture what that means in an ancient population. A mosquito bites two people, both become infected. The parasite multiplies in their blood. But one person has a lower probability of developing the severe form that blocks blood flow and damages vital organs. That person is more likely to survive. Maybe the difference is only a little on average. Evolution does not require every advantage to be dramatic. Repeated across hundreds or thousands of generations, small differences can alter the frequency of genetic variants. In that environment, O may have been part of a complicated survival landscape, but nature does not give free advantages. The same ABO biology associated with lower risk of severe malaria can be associated with greater severity of cholera. Cholera is caused by Vibrio cholerae, a bacterium that colonizes the intestine and produces a toxin capable of causing massive fluid loss. Several studies have reported that among people who become infected, those with blood group O are more likely to develop severe cholera than people with non-O groups. The exact molecular interactions are complicated, but the broad pattern is real enough to have become one of the classic examples of a possible evolutionary trade-off in ABO biology. Now the blood story becomes darker. Imagine a population exposed for thousands of years to both malaria and cholera. One blood group profile may reduce the chance of severe malaria but increase the risk of severe cholera. Another may have the opposite balance. Instead of one best blood type taking over, multiple alleles can remain in the population because the environment keeps changing the equation. That is the basic logic behind balancing selection. Researchers studying ABO evolution have found evidence that the gene carries unusually deep and complicated variation consistent with long-term selection. The A and B forms themselves have an extremely old evolutionary history, and different O lineages have arisen through separate genetic events. The system looks less like a ladder of progress and more like a battlefield where several genetic strategies have survived, because each can be useful under different conditions. And malaria is not the only infection that interacts with blood group biology. Norovirus, the virus behind many outbreaks of acute vomiting and diarrhea, can recognize carbohydrate structures on intestinal cells. Those structures are influenced not only by ABO type, but also by another gene called FUT2, which determines whether certain blood group-related antigens are expressed on mucosal surfaces. That means susceptibility varies by virus strain and by the interaction between several genes. This is precisely why dramatic claims like type O is immune to norovirus are misleading. The real science is messier and much more interesting. Blood group can influence infection biology without becoming a simple yes or no shield. There is another misconception that needs to disappear: the claim that O is simply the oldest blood type. It is easy to believe because the O cell lacks the A or B modifications and therefore looks like the simplest version. But simplicity is not the same as ancestry. The evolutionary relationships among ABO alleles are tangled. Different lineages have recombined. Some have undergone gene conversion. Different O variants have independent histories. Molecular studies have even proposed that some modern A-like sequences arose through recombination involving O and B backgrounds. Whether every detail of those evolutionary models remains accepted is a separate question, but the larger conclusion is secure. ABO evolution cannot be reduced to a straight sequence in which O came first, A came second, and B came third. Now look again at the phrase O positive. The O and the positive are not one genetic system. They describe two different blood group systems inherited independently. The O belongs to ABO. The positive refers primarily to the RHD antigen, the feature responsible for the familiar plus or minus designation. So O positive does not represent one ancient genetic package. It is a combination of an ABO phenotype and an Rh phenotype. The plus sign does not tell you that your ancestors came from a certain ancient region, and it does not make the blood biologically stronger. It is simply another inherited characteristic. There's also an important medical myth here. O positive is not the universal donor blood type. In emergency red cell transfusion, O negative is commonly used as the universal red cell donor because those cells lack A, B, and RHD antigens. O positive red cells are compatible with many Rh positive recipients, but not with everyone. Hospitals rely on proper blood typing and cross-matching because the ABO and Rh systems are only part of a much larger collection of blood group antigens that can matter clinically. But there's a beautiful twist in the story of the O allele. One of the common human O variants involves a single base deletion that effectively shuts down the enzyme. Researchers have shown that this particular deletion appears within ancient O lineages, but they have also documented other O alleles created through different mutations and recombination histories. The molecular record is telling us something profound. Evolution often reaches similar functional outcomes through different genetic routes. Several paths can produce the O phenotype. So the blood moving through your veins is not the result of one ancient accident copied unchanged forever. It is the living product of a family of genetic solutions. And some of those solutions became especially common because human populations repeatedly passed through bottlenecks. This is where migration enters the picture again. When a population becomes small, rare variants can suddenly become common simply because the surviving individuals happen to carry them. A later population explosion then multiplies those frequencies. That is one reason the genetic landscape of a population can look remarkably different from the population it came from. In the Americas, founder effects and later drift appear to have played a major role in producing the high frequency of O in many indigenous groups. In other places, disease and selection may have pushed the frequencies in different directions. The same gene is responding to a different history. This also explains why scientists are cautious when discussing ancient ancestry. Blood group frequencies are useful, but they are not fingerprints in the forensic sense. Two populations can have similar O frequencies for entirely different historical reasons. A gene may become common through selection in one population and through founder effects in another. The strongest conclusions come when ABO data agree with larger genomic studies, archaeological evidence, and known migration histories. The deeper lesson is that biology and history are never completely separate. An epidemic is history. Migration is history. A population bottleneck is history. A genetic mutation is history. And sometimes all four end up written into the same gene. That is why the O blood group has fascinated evolutionary geneticists for so long. The gene has persisted through ancient hominin populations. Different alleles have emerged and spread across continents. Disease has probably influenced its distribution. Founder effects have amplified it in isolated populations. And modern medicine depends on understanding the consequences of those tiny molecular differences. There's even a famous example that shows how weird ABO biology can become. In 1952, doctors in what is now Mumbai identified a patient whose red blood cells lacked the H antigen, the precursor structure on which A and B antigens are normally built. This phenotype is called the Bombay phenotype. A person with this blood type can appear to be type O in routine testing, but ordinary O blood is not necessarily compatible because the person lacks the H antigen itself. The phenotype is rare worldwide but occurs at higher frequency in parts of India. It is a reminder that what looks like a simple blood group can conceal deeper layers of genetics. You can appear to be O while carrying a completely different underlying biological situation. And now, imagine placing all of these pieces together. An ancient gene, multiple O alleles, a long evolutionary history, archaic humans carrying O-associated variation, founder effects in the Americas, potential protection against severe malaria, higher severity of cholera among some infected O individuals, norovirus interactions influenced by both ABO and FUT2, and a separate Rh system determining the positive sign. Suddenly, O positive does not look like a random label anymore, but it also does not look like a secret destiny. That distinction matters. Your blood type does not tell you whether you are genetically better, smarter, stronger, or more evolved. It does not tell you exactly where your family came from. It does not guarantee protection against any infection. And it should never be used to make simplistic claims about race or ancestry. Those conclusions go far beyond what science can support. The real story is much more impressive because it is about survival without perfection. Your ancestors did not carry an unbeatable blood type. They carried variation. Sometimes one variant helped, sometimes another hurts. The environment changed. A disease spread. The population shrank. A few survivors had children. A new population expanded. And millions of years of evolutionary history accumulated without anyone planning it. That is what genetics means by adaptation. It is not a designer. It is not a prediction. It is a record of what happened when living populations met changing environments. So the next time you see O positive written on a medical form, think beyond the hospital. Think about the molecular machinery inside chromosome 9. Think about the ancient O lineages that survived deep into human evolution. Think about populations entering the Americas with only a fraction of the genetic diversity available in Asia. Think about the possibility that malaria shaped some blood group frequencies while cholera pulled in the opposite direction. Think about the countless generations in which the same tiny molecular differences were tested by the environment over and over again. Your blood type is not a secret message from one mysterious ancestor. It is something bigger. It is a small surviving piece of an evolutionary argument that is never completely ended. And the darkest part is that there was never one answer. There were only trade-offs. A genetic feature could help against one enemy and hurt against another. The same body could carry biological advantages and vulnerabilities simultaneously. Evolution was not trying to make humans perfect. It was trying to keep populations alive long enough for the next generation to exist. That is why O does not mean best. It means one evolutionary solution, one of several, a solution that became common in some populations because history pushed it there. And if you are O positive, that is what your blood is carrying right now. Not a mystical secret, not proof of one forgotten tribe, but a living record of countless generations of mutation, migration, disease, chance, and survival. The letter on the chart is tiny. The history behind it is enormous. And the remarkable thing is that scientists are still reading it. Every new ancient genome, every newly characterized ABO variant, and every better study of infectious disease adds another piece. The story will almost certainly become more complicated, not less. We may discover populations that carried variants we never expected, selection pressures that changed blood group frequencies in ways we cannot yet explain, or ancient migrations that become visible through combinations of markers rather than any single gene. For now, one fact stands above the mythology. Type O is ancient. Its genetic history is diverse. Its distribution has been shaped by both chance and selection. And the reason it survived was never because nature found a perfect blood type. Nature never needed perfection. It only needed enough survivors. And somewhere in that enormous chain of survivors, generation after generation, a small genetic difference remained. It reached you. It is moving through your veins right now. And the ordinary little O positive on your medical chart is the modern label for a story that began long before anyone knew what DNA was, long before blood groups had names, and long before humans had words for the diseases that shaped them. That is the real secret in your blood. Not where one ancient person came from, but how many generations had to survive for you to be here at all.